Welcome to Geology 108, Origin of Life. The question of the origins of life has puzzled perhaps every single human being since the rise of consciousness. Life is ubiquitous around us, and yet how it came to be seems to elude simple explanations. Where do we come from? Why are we made this way? Is there life elsewhere in the universe? And what is life exactly? Addressing these fundamental questions has historically been the focus of philosophy, art, and religion, but only recently has the question of the origin of life come under the scrutiny of science. By following the pillars of the scientific method, hypothesis testing, and the weight of evidence, we can now discuss probable scenarios of how life came about on Earth. A proposed hypothesis for the origin of life on Earth is that it was brought from outer space, the so-called panspermia hypothesis. Earth receives much organic matter from outer space on a yearly basis. So this explanation is not excluded. However, it is not satisfying because it does not address how life would have started before its journey to Earth. The central question that we will discuss here is how life emerged from non-living matter in a pre-biotic world, in the conditions offered by early Earth of the Hadean and Archean eons. Better understanding of the process of biogenesis gives us clues to the likelihood of life existing elsewhere in the universe and may provide us with new insights into our place in this world. Life takes many forms and is materialized in a wide range of organisms made of a single cell or multi-cell symbiotic structures. We can point to many aspects of life that distinguish it from non-living matter, but as we seek to understand the birth of life in a prebiotic world, we focus on characteristics defined at the molecular level. At which point can we say that a collection of atoms and molecules is alive? Live organisms are a combination of intricate molecules with complex interactions that allow four critical functions. One, metabolism is the suite of chemical reactions that life forms use to sustain their activities. Two, growth involves the ordering and organizing of atoms and small molecules to make larger molecules by crystallization and polymerization. Three, reproduction is the self-duplication of molecules, carrying along genetic information from one generation to the next. Genetic material can mutate, providing genetic variations. And mutations can produce chemical novelty. And finally, four, evolution operates on multiple generations of live organisms as molecules incorporate new functions, optimize their efficiency, or develop symbiotic relationships with other molecules, allowing them to increase their chance of survival. A set of molecules that features all these characteristics can be said to be alive. Life operates through the specialized chemistry of carbon and water. Life also builds largely upon four key families of chemicals. Lipids are used to form cell membranes. Carbohydrates, like sugars and cellulose, are used to store energy and create rigid structures. Amino acids are used for protein metabolism. And nucleic acids, DNA and RNA, are used to store and execute genetic encoding. Any successful theory of biogenesis must explain the origins and interactions of these classes of molecules. To explain the origin of life, we must understand how the processes of growth, metabolism, reproduction, and evolution may operate at the molecular level in a pre-biotic world. These functions can operate in isolation. If they operate in isolation, life can appear when the molecules that carry out these elementary functions are grouped together and organized cooperatively. The transition from non-living to living entities was not a single event, but an evolutionary process of increasing complexity that involved molecular self-replication, self-assembly, autocatalysis, and the emergence of cell membranes. Although the details are still unknown, biogenesis must have proceeded in incremental steps, each with added molecular and chemical complexity. First, pre-biotic molecules, the building blocks of cells, must come to existence. Since all live organisms are built with organic matter, raw organic molecules must become widely available in a prebiotic Earth. Then, molecules will have to be selected, concentrated, and organized into the essential structures of life, a membrane-protected cell. Along the way, molecular functions should have included reproduction with passing of information. The mechanisms by which these processes take place is by natural selection, whereby molecules that attain advantageous functions survive over the competition. Molecular evolution was the constant thread leading to the emergence of life. The first and best understood step in biogenesis is the production of the molecular building blocks of life: sugars, amino acids, lipids, and more. Anywhere carbon dioxide and water interact, in the presence of a sustainable energy source, the building blocks of life do form. Lightning in the atmosphere, volcanic activity near the seafloor boiling the ocean, or even solar radiation in the molecular clouds of the solar nebula before Earth was born. Organic matter is made of the most abundant elements and is ubiquitous in the cosmos. Even currently, tons of organic material rain on Earth's surface from outer space every year. Early Earth had a carbon-rich secondary atmosphere and was covered with wide oceans. Any source of energy favored the creation of prebiotic molecules. The seas of ancient Earth became increasingly concentrated in biomolecules that rained from the skies and rose from the depth at hydrothermal vents. As water and carbon dioxide are widely available in the cosmos, the building blocks of life may presumably be found all around the solar system and the universe. The most famous experiment in biogenesis dates from 1953, when chemists Harold Urey and Stanley Miller designed a simple and elegant tabletop glass apparatus to simulate early Earth. Gently boiling water represented the hot Hadean Ocean. A mixture of simple gases mimicked Earth's primitive atmosphere. Electric sparks simulated lightning. After a few days, the water turned pinkish, then brown, with a complex mix of organic molecules, amino acids, and other biobuilding blocks. The Miller-Urey experiments demonstrated how prebiotic molecules could have formed spontaneously in the early Earth, creating the paradigm of the prebiotic soup as the origin of life. Earth must have abounded in the essential molecules of life. The Miller-Urey experiments do not reveal how the biobuilding blocks are combined to accomplish the other molecular functions of growth, metabolism, and replication that are critical for life. But early Earth offered a much wider variety of physical and chemical environments than captured in this experiment. In the late 1980s, the discovery of deep black smoker ecosystems gave rise to the viable alternative to the primordial soup. In those deep dark zones, far from the sunlit ocean surface, mineral-rich fluids interact with hot volcanic crust to generate ocean floor geyser-like vents. Jets of scalding water contact the frigid deep ocean to create a constant precipitation of minerals, the microscopic particles that produce the black smoke. Amino acids, lipids, and other biobuilding blocks also spontaneously form in black smokers' environments due to the heat circulated by fluids above hot magma and the presence of reactive mineral surfaces. Black smokers today are found on the seafloor at mid-ocean ridges, where magma cools down to create new oceanic crust. These conditions are representative of early Earth, where volcanic activity was intense underneath a global ocean. Life abounds in those hidden places, fueled by the chemical energy at the interface between crust and ocean. The existence of black smokers at undersea vents underscores that life abounds in extreme environments. We estimate that some 200 to 600 octillions microbes live in rocks beneath the continents and even more beneath the seafloor. Subterranean diversity rivals that of the surface, with most underground organisms yet to be discovered or characterized. These microorganisms extract energy and essential elements from rocks and water. Deep rock microbes only reproduce every few decades and enter a dormant stage when nutrients run low. The population of an entire community may take thousands of years to be replaced, and they may have been isolated from the surface for millions of years. If a significant fraction of Earth's life survives in deep environments protected from the violence of asteroids and comets and from the tempers of climate change, life could have originated there underneath the surface. This also implies that similar organisms may be found in many places in the solar system and beyond, fed by the chemical potential between deep and shallow minerals, bathed in the accretionary heat of planets and moons. Mars today must have deep hydrothermal zones. Perhaps life endures there even now. Several of Jupiter's moons are also ripe for biological investigation, as is Titan, Saturn's organic-rich, Earth-sized moon. Every ancient environment with sources of energy and small carbon-bearing molecules probably produced its share of amino acids, sugars, lipids, and other molecular building blocks of life. The secondary atmosphere, rich in carbon dioxide and water vapor, and exposed to lightning and solar radiation, probably rained pre-biotic molecules all around the planet. But while lightning may facilitate the production of small biomolecules, they can just as easily rip those building blocks to molecular shreds. In contrast, the protected and stable environment of submarine volcanic zones provided a constant supply of minerals and heat to produce these molecules and preserve them for long periods. Volcanic gases containing carbon and nitrogen readily react with common rocks and seawater to make virtually all of the building blocks of life. These synthesis processes are governed by relatively gentle chemical reactions called reduction and oxidation reactions, collectively called redox reactions. such as the familiar rusting of iron or caramelizing of sugar. These are the same kind of chemical reactions that life uses in metabolism. Hence, the submarine environment was probably the most auspicious place to birth life. But biomolecules also form in deep space molecular clouds exposed to cosmic rays. So the building blocks of life were and still are abundant everywhere in the cosmos. The remaining question is how this material was concentrated and organized to provide function. During the Hadean, a period of intense submarine volcanic activity, it was easy to make amino acids, lipids, and sugars from a combination of water, carbon dioxide, and minerals in volcanic zones, protected and submerged by a global ocean. Indeed, the problem is making too many different kinds. A fundamental attribute of life is chemical parsimony. Only a few small molecules are used by life, but nature manufactures millions. The second stage in the origins of life was the selection, concentration, and assembly of molecules from the complex but dilute prebiotic soup into useful macromolecules. Those macromolecules include structure that enclose the cell, promote its chemical reactions, and carry genetic information. Two complementary processes likely played a role: lipid self-assembly and template-directed synthesis. Many sources of biomolecules form lipid molecules that self-assemble in water. Lipids in water can spontaneously organize into tiny spheres the size of a cell because one molecular end is attracted to water and the other is repelled by it. These molecules can find each other and form a double layer with water-hating ends pointing inward, away from the water, and water-loving ends pointing outward, in contact with water. Lipid self-assembly must have played a key role in the origin of life. However, most of the other biological molecules don't self-organize. Sugars and amino acids, for example, tend to dissolve in water and remain there in solution. Instead, these molecules can become concentrated and neatly arrayed on the protective surface of minerals. Some biomolecules carry a slightly positive electric charge, while others have a slightly negative charge. Other few molecules, like water, are polar, meaning that they have both slightly positive and negative ends to the same molecule. In a similar fashion, minerals have charged surfaces. Some mineral surfaces are slightly positive and some are slightly negative. And sometimes a mineral will have both positive and negative regions on its surface. The result of these charged regions of molecules and surfaces is that they can organize spontaneously as positive electric charge attracts negative electric charge. Template-directed selection on mineral surfaces of different shapes and electrical charges has concentrated and selected specific biomolecules. Chemical reactions at the interface between water and minerals also provided a source of energy for further transformation of biomolecules concentrated on the seafloor. All sorts of molecular interactions further refined the prebiotic mix. Some groups of molecules cooperated with each other to attach to various mineral surfaces in ways that individual molecules could not, thus enhancing survival of the group. Other small molecules must have acted as catalysts that enhance survival in two ways. Some chemical species promoted stability by catalyzing the formation of new chemical bonds, while others accelerated the destruction of molecular species by breaking their bonds. In this way, the molecular soup was inexorably refined. Perhaps the most daunting challenge in molecular selection is chirality, the pervasive handedness of life. Many life's molecules come in mirror image pairs, left and right-handed variants, like your two hands. Chiral pairs of molecules are identical in many respects. They have the same chemical compositions, the same boiling and melting points, the same color and density, and the same electrical conductivity. But left and right-handed molecules have different, incompatible shapes. It turns out that life is incredibly picky. Cells almost exclusively employ left-handed amino acids and right-handed sugars. Chirality matters. In the case of the artificial fragrance limonene, the right-handed form smells like an orange, whereas the left-handed version of this simple ring-shaped molecule smells like a lemon. The smell receptors in your nose are sensitive to chirality, so right and left limonene transmit slightly different signals to your brain. Taste buds are less sensitive to the differences between right and left-handed sugars. They both taste sweet, but our body's fine-tuned digestive system can process only the right-handed forms. The artificial sweetener tagatose, a zero-calorie left-handed sugar substitute, exploits these properties. The tragic story of thalidomide also rests on handedness. The right-handed version of this drug alleviated morning sickness in pregnant women, but the left-handed variant that inevitably tagged along caused birth defects. The non-living natural world is for the most part indifferent to the distinction between left and right molecules. But life absolutely requires the correct shape. Left-handed amino acids and right-handed sugars are essential. The commonest minerals in every rock abound with surfaces where atoms form molecular scale handholds, some left and some right. These left and right-handed mineral surfaces occur in statistically equal proportions. So Earth, on a global scale, doesn't appear to be biased for either left or right. But each individual molecule does care where it winds up. Certain left-handed molecules can aggregate on one set of crystal structures, while the mirror image, right-handed counterpart molecules, aggregate just as easily on other sets of mineral surfaces. Take countless trillions of mineral surfaces, bathed in a wide range of pre-biotic molecules. Repeat those tiny natural experiments over and over for hundreds of millions of years. And Earth must have eventually tested virtually every combination of small molecules somewhere, sometime. The molecules that learned to replicate quickly took over the world, as the competition could not produce randomly at the same pace. If these molecules developed mineral surfaces that are compatible with only one side of amino acids, this preference was inherited by all descendants, becoming the trademark of all subsequent live organisms. The origin of life required that simple molecules assemble into the essential macromolecules of biology, including cell membranes, proteins, and the nucleic acids, DNA and RNA. Ultimately, for life to get started, collection of molecules must learn to self-replicate. Three rather different models are in competition to describe that first self-replicating system. The reverse citric acid cycle, autocatalytic networks, and self-replicating RNA. By far the simplest of these three models is based on the well-known cycle of a few small molecules called the citric acid cycle. The idea is to build up bigger molecules from smaller pieces. Start with acetic acid, which has only 2 carbon atoms, and react the acetic acid with carbon dioxide to form pyruvic acid with 3 carbon atoms. Then pyruvic acid undergoes a second reaction with carbon dioxide to make oxaloacetic acid with a backbone of 4 carbon atoms. These initial reactions are followed by other simple chemical reactions that produce progressively larger molecules up to citric acid, which has 6 carbon atoms. This cycle becomes self-replicating because the citric acid molecule splits into 2 pieces, 2 smaller molecules, acetic acid and oxaloacetic acid. But these 2 smaller molecules are both part of the original cycle. So now you have 2 cycles going, one starting with acetic acid and the other with oxaloacetic acid. In that way, 1 cycle of molecules becomes 2 and 2 cycles become 4 and so on. It turns out that it's easy to synthesize many of life's essential building blocks, including amino acids and sugars, by simple chemical reactions with the core molecules of the citric acid cycle. One of the most convincing arguments in favor of the citric acid cycle as the first self-replicating system is that it is very ancient. Just about every known living cell on Earth has the citric acid cycle buried deep inside. So it is a primordial characteristic that may have descended from the very first life form. Another scenario of self-replication is the RNA world, a model based on a hypothetical molecule of RNA that makes copies of itself. This scenario is appealing because it combines 2 critical functions, metabolism and genetics. Modern cells use DNA to store and copy the information needed to make more proteins, but they use complexly folded proteins molecules to make the DNA. So which came first, DNA or proteins? In fact, RNA plays a central role in both processes. RNA is a long, single-stranded molecule assembled from smaller individual molecules called nucleotides, like beads on a string or letters in a sentence. 4 different molecular letters designated as A, C, G, and U can line up in any sequence, like a coded message. These RNA letters hold genetic information just like DNA. At the same time, RNA can fold up into complex shapes that have the ability to catalyze biological reactions. The RNA world model rests on the assumption that some chemical mechanisms produced vast numbers of different strands of RNA, and perhaps an information-rich molecule very similar to it. Almost all those varied strands did absolutely nothing. They simply survived or gradually degraded. However, a few select strands possess some kind of self-beneficial function. They folded up to become more stable, or they latched on to some secure mineral surface, or perhaps they destroyed their rivals. Just another example of molecular competition. The key assumption of the RNA world hypothesis is that one of these myriad strands learned how to make copies of itself. It became a self-replicating molecule. RNA is also easily mutated, so the self-replicating RNA molecule quickly found itself competing with lots of slight variants of itself. This RNA molecule would fulfill all of the requirements of life, a self-sustaining chemical system capable of incorporating novelty and undergoing Darwinian evolution. In this case, molecular evolution. In the prebiotic broth, a collection of molecules with even the slightest useful function had an advantage. But an RNA strain could make copies of itself. Such a self-replicating molecule ensured its own survival by producing more or less identical offspring. What's more, the molecular copying process was inevitably messy. So some of those RNA copies were mutants. And while most mutations were lethal or conferred no significant advantage, a few fortuitous individuals outshone their parents, and thus the system evolved. Simply by chance, copying errors, the original self-replicating molecule must have produced offspring that tolerated more extreme conditions of pressure or heat or salt, or replicated faster, or found new sources of food. Even greater advantages were enjoyed by those RNA strands that found protection on a mineral surface or refuge within a safe encapsulating membrane. Without competition, the first self-replicating molecules spread around Earth in a geological instant. For 500 million years, cells fed on chemical energy available in their environment by chemosynthesis. The culmination of the innovation process in the Archean Eon was the invention of photosynthesis, the discovery of a far greater source of energy, the sun. However, early photosynthesis did not produce oxygen as a byproduct. Life originated on Earth when the first living cell began to replicate, sometime before 3.5 billion years ago. For the next billion years, single-celled life radiated and diversified, but appears to have had little effect on Earth's near-surface environment, including its rocks and minerals. Life was concentrated in the ocean because destructive ultraviolet radiations inundated the land. For a billion years or more after the innovation of that first living microbe, Earth's surface hadn't changed in any significant way. New varieties of microbes probably formed brownish scum along some coastlines. A few pioneering cells began to experiment with ways to capture the sun's energy. Everything changed when cells learned to release a highly toxic, highly reactive gas, oxygen. Photosynthesis provided Earth with its tertiary atmosphere, one rich in oxygen with only trace amounts of carbon dioxide. This transformation allowed life to conquer the land and to develop more complex life forms.